Hello, I'm Randy Bateman, and I'm presenting today on updates on blood and CSF biomarkers, as well as the DIAN-TU clinical trial status. On this slide are shown my disclosures, including my research-supported initiatives, as well as relevant to this discussion of Biomarkers, a company that Dave Holtzman and I co-founded, C2N Diagnostics. Alzheimer's disease has been an enormous challenge for us as a modern society where many of us are expecting to live into our later years. It is the most common form of dementia. It's commonly seen with comorbidities and copathologies. Previously, there has been no cure and no disease-modifying treatments. However, I'll show recently where there's very good news that treatments can change the course of this previously universally fatal disease. There's enormous impact on the individual patient, the family members, and society, both financially, but also just from the numbers of people who are suffering with the disease and having to live with it. The costs are enormous, and recent advancements in science promise to change the course of this disease so that one day it may actually be something that is much less damaging to people. The history of Alzheimer's disease is summarized here. And since the initial description in 1906 of a pre-senile young onset dementia, not due to late onset senile dementia, the pathology was characterized with plaques and tangles. And since that time, the developments have really been accelerating, starting in the 1960s with pathology and clinical correlations, and the realization in the 70s that in fact, late onset senile dementia is Alzheimer's disease pathologically. And from that time, the race was on to identify what the proteins that comprise the plaques and the tangles are. And some of the first drugs were being developed based on neurotransmitter theories of the disease, the acetylcholine. And so these acetylcholinesterase inhibitors and memantine were all developed and approved in the late 1990s and early 2000s. During the 1990s, our biochemical ability to identify the changes in Alzheimer's disease were developed in cerebrospinal fluid for amyloid and tau, and later imaging abilities to measure pathology by PET scans. First amyloid and later tau PET scans were developed. And then I'll review most recently the development of blood-based biomarkers. But there's been some exciting developments in clinical trials of Alzheimer's disease, where, as I mentioned, the certainty of the course of this disease has really changed. And so these medications all treat the symptoms and typically do not change the course of the disease, but have some added benefit in cognition, which usually isn't noticed at the individual patient or family level. However, recent trials in 2020, 21, and now in 22 have shown that not only can the disease and the pathology be reversed and plaques removed, but with plaque removal, multiple monoclonal antibodies are now showing that we can change the course of progression in cognitive, clinical, and functional outcomes of the disease. So this is really a transformational time in our field. This has never been possible before. And the disease has been with us for thousands of years, probably for as long as we've been humans. And the ability to change this course is really a notable chapter in the development against treatments for Alzheimer's. I'm going to be talking a lot about a rare form of Alzheimer's, dominantly inherited Alzheimer's disease, and I'm going to contrast and compare that to sporadic Alzheimer's disease. The dominantly inherited form typically starts at a much younger age of onset, and we can know ahead of time exactly what the etiology is, mutations in genes that cause the disease. Whereas in sporadic AD, as the name suggests, we don't know the precise etiologic cause, but we do know the pathophysiology, pathology, and the associated biochemical changes. What you'll see is in this comparison table, the clinical cognitive presentations, the MRI atrophy, the pathology by PET scans, hypometabolism, biochemical changes in CSF and blood are all largely similar or the same between these two forms of the disease. There are some differences. For example, in the early onset mutation carrying form, typically those individuals will have more pathology, amyloid and tau pathology, and less other comorbid pathologies. They will also have less comorbid disease. The first case, it turns out, of Alzheimer's disease, Auguste Dieter, her brain tissue was analyzed and found to potentially have a mutation that caused her disease. And since the early 1990s, multiple mutations have been identified and described that cause autosomal dominant Alzheimer's disease in families. Now, this is very rare, but when it strikes in a family, it's very common. Half the people in the family are at risk of getting the mutation, and every person that gets that mutation will go on to get Alzheimer's disease with incredibly rare exception. And so these genes, amyloid precursor protein, presenil 1 and presenil 2, are all involved in amyloid beta processing. And this led to the amyloid hypothesis, which has been developed for several decades. And the question was, how does the change in the disease, how does it progress in a way that leads from the first mutation, leads through that pathophysiological process to dementia? And in order to help address this question, the NIH established the Dominantly Inherited Alzheimer Network, which is a group of centers all around the world working together, trying to describe the changes that occur from that first mutation when it's inherited all the way through that life process to full-blown dementia. And so the order and rate of these changes is critically important to understanding the disease. The centers are shown here. This includes the observational studies and the trial sites colored in different, but you see North America, Central, South America, Europe, Asia, and Australia are all members, and there's other centers that are becoming involved at different levels. One of the first realizations of the ability of these mutations to predict what's going to happen was the study of the age of onset. And so it turns out that the age at which a parent has the onset of their clinical symptoms is related to the age at which their descendants can expect to have their age of onset. And when you average by mutation all the ages of onset here on the x-axis, you can predict to a fair degree of certainty approximately when individuals might have their clinical age of onset. And we can use this as an estimate then, so that if someone is 10 years before their expected age of onset by their mutation, we could call that estimated years to symptom onset of -10. And we can use this to track them the process many years before the symptom onset. Shown here is the amyloid PET scans on the left and the top, and you see substantial amounts of amyloid plaques depositing 15 to 20 years before. And then at about 7 years before, we have the hypometabolism setting in the brain, followed at about 5 years before symptom onset of atrophy in those same regions. Now, what we know now is that tau tangles right at symptom onset also develop right in these same regions where the hypometabolism and the atrophy are occurring. So all of this indicates a sequence of events starting some 20 years before symptom onset in these mutation carriers that lead up to symptom onset with tau tangles. Now, to emphasize this point, you can look at tau PET scans, the aggregated form of tangles that can be measured by PET scans. And what's remarkable is that all the mutation carriers shown in yellow triangles have completely normal levels of tau pathology, with the exception of one, if they are asymptomatic. But the year they become symptomatic, all of them have elevated levels of tau PET, with the exception of one. And so this is almost a perfect binary detector of clinical symptom onset. And what this indicates to us is that the tau pathology that we measure by PET scans is very closely tied to the symptom onset of dementia. Here you can see the bar chart and scatter plots. So how do we put all this together? These are the actual quantitative Z-score curves. For example, amyloid beta deposition. This is PET scan. You can see CSF tau, CDR sum of boxes. This is a stylized tau PET curve, and hypometabolism and atrophy is shown here. And you can identify now that if this is the tangle growth stage of the disease, that's the symptomatic phase. And this is where most clinical trials to date for Alzheimer's have been done. If you look at the 15 to 20 years before, this is the plaque growth stage. This is when the amyloid plaques are really substantially growing. But as I showed, once symptom onset occurs, the plaques are fully established. And then there's the pre-pathological stage where we don't detect plaques, tangles, or other pathology, even though there may be biochemical abnormalities due to the mutation. And we refer to that as the primary prevention, meaning before pathology, secondary prevention when pathology has begun, and the symptomatic phase for our trials that I'll talk about. So for diagnostic development, when I was a resident, we were taught you could only diagnose Alzheimer's disease with a brain biopsy or after someone had died, brain autopsy. And that hasn't been true for a couple of decades now. And I want to emphasize this point because since the 1990s, we had pretty good CSF biomarkers. Then in the 2000s, PET scans were developed for amyloid and tau PET. And we really have the ability to quite accurately diagnose Alzheimer's disease in life and really at almost any stage. I showed up to 15 to 20 years before the changes occur. And the latest breakthrough really came in the past few years when blood biomarkers have been discovered and developed to identify these same kinds of protein changes that we detect in CSF. We can now quantify in blood to a highly accurate degree. And so we have PETs, we have imaging, and we have CSF, and now we have blood. And each of these can accurately identify and specifically identify the pathology of Alzheimer's disease. And as I'll show you, that accuracy rivals that really of any clinical test that we have in neurology and medicine. And so there really is no reason to talk about brain biopsies as the only way to diagnose Alzheimer's disease. Our standards certainly meet that of medicine. And so in support of this, the FDA has given approval, for example, of PET tracers and CSF, and we think likely soon blood tests will be approved by regulatory and payers to use in the clinic. As part of the development of this, though, there is an enormous amount of science that goes into this. And one of the ways to analyze proteins, for example, that cause disease is to look at how they're made, produced, transported, cleared, what forms they take. And one way to do this is a method we developed called stable isotope labeling kinetics. And this allows one to measure the turnover rate of proteins in models or in people and how long. And so some of our proteins have been with us for a very long time, our whole lives, collagen and crystalline in the eye and other things. And other proteins have turnover rates of a year or months or days. And some proteins like amyloid beta and ApoE, they have a turnover rate of hours. So a person will make and clear their amyloid beta in their central nervous system and their ApoE on the order of 8 to 12 hours. And it's really quite remarkable when you think about it. So during an average day, you'll make and clear half the amyloid beta in your brains, and over a day and a half, 2 days, all of it will be turned over, unless you're accumulating amyloid plaques, in which case ApoE and A beta deposit in those. And tau is 23 days for its average half-life, and the same is true for that. So tau is remarkably turned over at a fairly rapid rate when you think of that it's a structurally associated protein that's holding your microtubules together, and it's part of your nervous system. There is a pretty rapid turnover of that. It's not like collagen or crystalline that sits there for your whole life. And so one can do this in cell culture or in mice. We use mass spectrometry to detect this label in kinetics. Most of our studies are in humans where we infuse the label and sample the blood and the CSF over time. And as we sought to understand the economy of amyloid beta, where is it made, where is it going, what is it doing, we developed studies to measure this transport. And one of the transport experiments is an AV experiment, arterial to venous transport experiment. And what we found was that there was a specific transport from the central nervous system into the blood of amyloid beta, and that it's cleared in different ways, but one of the ways is active transport into the blood. And we estimate that about half of the amyloid beta made gets put into the bloodstream. And as we did this, we tracked the kinetics, and what we found was just like in CSF, those people with amyloid plaques had disturbed amyloid beta 42, shown in red, relative to 40, compared to people who did not have plaques, shown in blue. And so this characteristic kinetic curve indicates that there's an imbalance in the amyloid beta 42 relative to 40 transport. And with this information, we began to explore what could we detect in the blood then of amyloid beta. And what we found in these initial studies was that amyloid beta 42 to 40 in the blood is pretty specifically lower, just as it is in CSF in people with amyloid plaques, shown here, compared to people without amyloid plaques. And that was regardless of their cognitive status, whether they had clinical dementia or not, shown in triangles and circles. We also found that if you use ApoE4 as a covariate in the analysis, you have better separation between the two groups because it accounts for some of the potential interactions in the blood. And so we had completed 3 studies from national studies, ADNI in the US, ABLE in Australia, and BioFinder in Sweden. And what we found was that across all these studies with different collection protocols and sample handling protocols and populations and cohorts, the cutoff was remarkably similar across these 3 studies for the 42 to 40 ratio. And they gave similar performance area under the curves of about 0.83 to 0.87, which really is quite good, again, for a detecting test, because in the clinic, in primary care clinics, approximately half the people are misdiagnosed or not diagnosed accurately with Alzheimer's disease. And so obviously 85% is better than 50%. And again, adding ApoE improves these by a few percentage points. Similar to CSF in blood, we found that, this was work, by the way, led by Suzanne Schindler. We found in our Alzheimer's disease cohort at WashU that there is a conversion from amyloid negative to amyloid positive in people who have abnormal blood plasma markers. So all these people start at PET negative, but then on average, by about 5 years out, half of them, and if we follow the data out, we predict it'll be everybody, converts from PET negative to PET positive. So this marker appears to precede our ability to detect it by PET, probably because it's a soluble change that's occurring that then consequently leads to the accumulation of plaques in the brain. One important caveat in the use of this and all other biomarkers is that we have to keep in mind what is the performance characteristics of the assay. And the reason that's important is because that can change our biological interpretation. So if a biomarker has high variance or low sensitivity, it can actually misinform about the order of changes in the disease process. Conversely, biomarkers that have very high sensitivity and high precision compared to other modalities that have a different level of precision can again misinform about the order of events, the magnitude of events, the association with other cofactors. And so several comparative studies have been done, and these are useful to look at to interpret so that we don't misinterpret the biological answers with something that is really a technical or analytical issue. One of the most exciting things right now that biomarkers are being used for is to accelerate treatment development in Alzheimer's disease. So many trials today are now incorporating blood-based biomarkers. And by incorporating these biomarkers, the numbers needed to be pre-screened can drop substantially, and the time it takes to enroll a trial through the screening process also drops substantially. And that the most important thing is that that saves time. That starts our trial sooner and it ends our trial sooner, giving us an answer sooner. And that will translate into more treatments and faster treatments for patients. And this can be quite substantial. For example, the average clinical trial for a symptomatic trial may take 2 years to enroll. For prevention trials, they can take 3 to 4 years to enroll. And to identify people, you have to screen 6, 7, 8 people to have one person come into the trial. But if you can pre-screen large numbers of people with blood tests, you can cut off years of that study, and that's a substantial effect. I'm going to switch gears now and talk about tau. So there are 4 points that I think are important to understand about soluble tau biomarkers and their relationship to the disease process and how to use them and interpret them. One is that soluble tau is very different from aggregated tau, and I'll emphasize that. Two is that different tau species, for example, different phosphorylated species, different fragments, really represent different processes, and that these different processes cannot just be different, but they can move in opposite directions. So we can no longer say that tau is hyperphosphorylated and sweep it under one generalization, because some go up and some go down. Tau site phosphorylation ratios, or the percent occupancy of a phosphocyte, is not the same as the total concentration of phosphorylated tau. And I'll talk a bit more about that. But that's important when we think about the biology and we begin to think about the drugs and the targets and how we're trying to intervene in the disease process. And then, as I just mentioned for the amyloid test, tau assay differences and sensitivity, specificity, and what they're measuring are important to keep in mind when we interpret the results of our studies. So one of the interesting things about tau is that most tau, until recently, that has been measured in fluids has been this N-terminal half of tau. And so what we know is it starts at the N-terminal and goes to about the mid-domain before the 4-repeat region. And this is where a lot of the phosphorylations that we track are at: 181, 217, 231, are all in this range. But this is what's almost all immunoassays to date have been measuring. Now, the other half of tau has not really been tracked in the fluid as much, but this is the half that actually becomes the tangles. And so it aggregates and makes pre-tangles and matures into neurofibrillary tangles. And you can see it's comprised of this 4-repeat domain. So structurally, the part we're measuring this in the fluids is structurally quite different than that which exists as the aggregated forms in the brain. And this is important. So recently, and I'll cover this data, we have been able to identify some of these C-terminal half of the proteins in biofluids and quantify those and understand better that relationship. So this is meant to highlight the fact that not all p-tau species are the same. For example, some measures of p-tau 181, 217, 231 are highly sensitive and specific markers for amyloid plaques. But when you look at other things like atrophy, shown here in cortical thickness, or in hypometabolism, shown by FDG PET, you see correlations with different p-tau species, for example, p-tau 205, and much less so with the other typical 217, 181, or the total tau markers. This C-terminal half, the microtubule binding region of tau, can be measured, and Kanta Hori has developed several assays with our group looking at this. We're using this now to track not just Alzheimer's disease tauopathies, but other tauopathies, and characterizing those in this region in the fluid that exists. But one particular region here, microtubule tau 243, seems to correlate very well with clinical status, and also remarkably with tau PET SUVR. And this appears to be an independent factor from the amyloid, the presence of amyloid, and is quantitatively quite precise, even at higher tau PET levels. So I think this is telling us something about this region and tau pathology and potential ways to track it without interacting with amyloid. So I mentioned that some markers, such as 217, are great markers for amyloid plaques, and Chihiro Sato has extended this work into looking at the CSF levels of 217 compared to A beta 42 to 40, and you see across many different diseases: cortical basilar syndrome, PSP, FTDs, tau mutations. You can see that there's a very nice relationship between phospho-tau 217 and the 42 to 40 ratio of amyloid beta, in exactly the way you would expect, with some notable exceptions. So, for example, this is amyloid negative in this quadrant, so the p-tau 217 is low, but as people have more and more amyloid, the p-tau 217 goes up. What's interesting is that some of these exceptions are actually in the tau mutations. So, for example, R406W remarkably has elevated p-tau 217. It's one of the few examples where you have elevated 217 without amyloid plaques. And so I think that's very important for a physiologic link between amyloid and tau. And I'm waiting for the day that a fantastic model is created using R406W. The performance of these metrics are shown here. This gets back to the percent that's phosphorylated. You see 217 in a sporadic AD group has a fantastic AUC for amyloid positive or negative, 0.97. We found the same thing in DIAN, 0.97. And so these phosphorylation ratios are really quite precise, 0.91 or 0.89, but not all of them. 202, for example, has a very low relationship with amyloid plaques. So again, not all phospho-tau species are the same. And the total concentrations also can tell us something different than the percent of phosphorylation at a site. So for example, here is a comparison done by Oscar Hansen's group and recently published looking at different plasma p-tau measures for 217 and 181 and compared to immunoassays and their sensitivity for detecting amyloid plaques. And you'll see that the percent phosphorylated tau done at 217 by our assay, by mass spectrometry, is one of the best performers for detecting amyloid plaques, but probably because it's actually looking at specifically the phosphorylation at that site and not the total tau concentration, which the immunoassays are currently measuring. And so this is really a percent of that phosphorylation. And the mass spectrometry also offers some specificity, but I really think it's this phosphorylation at that site that's important. And so now I'm going to cover a third class of biomarkers. Neurofilament light chain is a measure of neurodegeneration. And this goes up, it's not specific, it goes up in many disease states as shown here. But what's interesting is that it really begins to increase even before symptom onset. And so unlike the tau tangles we measure by PET, it suggests, shown here in DIAN, it's about 7 years before NFL really begins to increase. You can see the rates of increase in this plot and the converters and symptomatic mutation carriers. So these are people who they had high levels when they came in, but they weren't yet symptomatic. So this might be a proximal detector of the imminent onset of dementia in someone, say, that has plaques before they have tangles. And this is important because as we understand this protein, we need to understand more about it. What is it doing? We recently reported that there are fragments to NFL that exist in CSF just as there are for tau. And we have an NFL consortia to understand the biology of this and what these different regions mean. And do they tell us something different, each region by different diseases or by different disease processes? And shown here, you can see some regions are informative and others are less informative for Alzheimer's disease versus controls. So if we put all this together and we say that tau tangles really begin at the onset of dementia symptoms in an individual and progress through, and we look back, total tau increases some years before that. NFL, as I just showed, increases before that. P-tau 205 is increasing around the same time. So these may be in some kind of related process. And then the earliest changes, p-tau 181, 217, seem to be the earliest soluble phospho-tau changes and occur probably within a few years of the amyloid beta changes in 42 to 40 that leads to the aggregation. So these processes are much more entwined, and we're getting a very fine resolution now of the sequence of events that occur year by year leading up to dementia onset and progression. There are a lot of questions still to address, a lot of things we don't know, such as what do other disease states or comorbidities, how do they affect our biological measures of Alzheimer's? What processes the biology of what's happening to tau and NFL and other forms? What do those processes tell us about the disease? And these are major clues to what's actually happening. And that opens up the opportunity for potentially new targets to try to target the disease to slow and stop it. And then finally, we're dealing with the issue of how do we use these biomarkers? How do we use them in trials? How do we use them in the clinic to diagnose people? How do we use them to interpret what drugs are doing, how long we should treat for, or what the effects are? So I'm going to end this part of the talk giving acknowledgement to all the people who've contributed to these research studies, the participants and their families, the many researchers that have been involved in this, and in my lab shown here in the photo. We've recently established a Silk Center. I'm fond of this logo because it's glowing and moving, but we have opportunities for trainees such as postdocs. If you're interested in coming and doing this work, contact me. And there's many exciting projects and many things happening now. So now to shift gear, we're going to talk about treatment and what's happening in that space. Shown here is a map, an estimate of the different kinds of targets. You'll notice that we included exercise and diet and combination therapy in this because that's a really important part of Alzheimer's risk and progression. Now, the drug targets, what we typically think of, is shown in this inner ring. And you can still see that a substantial portion are based on amyloid beta, but not the majority. In fact, inflammation, vasculature, tau, ApoE, neurotransmitter, many other targets are present on this inner ring. And that's good. Diversifying our targets at this stage is important. We want to focus on amyloid. We've had great success there, but we want to see what else can be done. And so we're all familiar with the challenges of disease modification. What stage do we treat? How much do we treat with? And the first question is which target? I think this can only be answered with the completion of clinical trials. And we've been doing a good job of that. The field has been addressing this and making incredible progress along the way. And so just to, again, highlight, this accelerating development through the molecular biological understanding of the disease, building models, using those models to develop new therapeutics, testing those therapeutics, learning how to do it, getting the dose right, getting the safety profiles right, getting the stage of disease, getting the population right. All that has led to our most recent successes in drugs that are changing the course of the disease. But there's much more to be done, and I'm going to talk about going earlier and combination trials and how that can help. But in order to do that, it takes many stakeholders in a worldwide effort to accomplish these kinds of efforts. And shown here are the many groups, pharmaceutical companies, patient advocacy groups, government funding agencies like the NIH and other commercial supporters of the trials, the dominantly inherited Alzheimer network trials. Our first trials targeted amyloid beta. So we targeted plaques with a monoclonal antibody, gantenerumab, and soluble amyloid beta with solanezumab, and started beta secretase inhibitor trials. And shown here is the trial of the two antibodies that were started in 2012 and finished in 2019. And people who had mutations were assigned to one drug, the other drug, or a pooled placebo group, where we also use the observational data. And this is the summary of the trials, but I just want to highlight a few things. One is that gantenerumab really substantially changed the biological profile of these individuals. You can see in blue that it normalized the 42/40 ratio by 42%. CSF total tau, that N-terminal fragment we talked about, normalized that by 30%. p-tau 181 normalized that by 30%. And it even improved somewhat the neurofilament light chain by about 11%, a small margin. But look what happens when someone is asymptomatic. Before they have symptoms, this person has a head full of amyloid plaques, but over the treatment of the 4 years, completely removed their amyloid plaques. Someone who is symptomatic also had a head full of amyloid plaques, more, and during the 4 years of their treatment, they had substantial reductions, but not completely normal. And there's reason to believe that we need to reach that threshold to have a clinical cognitive impact. And so this next generation platform is launching out, incorporating tau with amyloid in combination trials. And we're doing this to have the biggest effect. And we're separating out our populations into those 3 stages I talked about: the symptomatic stage, the secondary prevention stage, and the primary prevention stage. And each of these is being treated differently. I'll show you that. But for the symptomatic and the secondary prevention, we're bringing in different tau targets and different tau drugs to try to test each of these in a diversified portfolio. So What is the rationale for tau? Well, I showed you that tau is the strongest association between clinical symptoms and AD pathology that we have. We also have strong scientific evidence that targeting both mechanisms, amyloid with tau, at the same time in the same person is more likely to have an effect than doing either one alone, because these two pathologies interact with each other, likely in a causal way. So our trial has just launched, and it's a combination trial of an anti-tau drug, a monoclonal antibody called E2814 by Eisai, and an amyloid drug, lecanemab, also by Eisai. And you'll see that the symptomatic individual arms will receive the anti-amyloid treatment first for 6 months, then be randomized to tau or placebo. Conversely, the asymptomatic people, the people who don't yet have dementia, will be randomized to the tau drug for 1 year, and then the anti-amyloid drug will be added on top of that. So combination of A and B together for both arms, but A starts first or B starts first, depending on the arm they're in. And then finally, the primary prevention trial is what we call the ultimate test of the amyloid hypothesis. This is led by Eric McDade, and we're very excited. This is now launched as well. It'll use gantenerumab to try to prevent amyloid plaques from forming in these individuals at the very earliest stages. Some of these people, this will be up to 25 years before symptom onset, so 25 to 15 years before symptom onset, and some of these people will be as young as 18 years old. So it'll be a real, what we think of as a truer prevention trial. And we believe that if we can prevent those plaques from forming, that it will interrupt the entire downstream process. So shown here by years is the DIAN-TU platform where we're at. We're currently in an open label extension of Gantt. We've had a cognitive run-in period that has now launched into this combination arm, and now the primary prevention trial. And we'll be adding more tau drugs in the near future. And so if people know of exciting new tau drugs or mechanisms, please contact us. I'm going to end by just communicating that this really is possible. I think the recent clinical results confirm that we can change the course of the disease. All the other treatments that remove amyloid plaques are showing this. But I want to show this example of a case where a young woman, in this case, she had cholesterol deposits in her skin that resolved over several months of being treated with the first HMG-CoA reductase inhibitor, a statin. These people now have decades of life added to their lifespan and healthy life due to cholesterol-lowering treatments like this. And by similar fashion, shown here again are these images in our gantenerumab trial. But if we can remove the plaques before people get sick, it's possible that we could have a quite substantial effect on their onset and progression. So with that, I'll leave this slide up here for resources, their websites and emails and phone numbers that can be contacted if people know if family members are interested in collaborations in the research study. And I'll thank the many members that it takes to do these DIAN and DIAN-TU studies, many of our advisors and performance sites around the world, as well as the DIAN steering committee. So all the sites direct the study in the directions that it goes. And the many cores that support it, our financial supporters and our trial sites are shown here. Thank you.